So good morning, everyone. So first, I would like to thank the meeting organizer for this opportunity to present Fujifilm's platform production process. So Fujifilm Diathin Biotechnology, stands for FDB, is a world-leading CDMO with over 25 years experience in CDMO experience of biologicals. So currently we have 3 operational sites, Bellingham, UK, and Research Triangle Park at North Carolina. Both sites specialize in recombinant protein expression from both microbial and mammalian cell expression systems. So our College Station, Texas site specializes in GMP manufacturing of viral vector for gene therapy, as well as for viral vaccines. So our company has completed over 310 projects, both in process development and on manufacturing space. So among these programs, we have completed so far 26 programs with process characterization and process validation. So as of now, companies support 6 licensed products for commercial manufacturing. So there are the 2 advanced therapeutical GMP manufacturing facilities currently up running at our Texas facilities. National Centers for Therapeutical Manufacturing, stand for NCTM, facility is designed to provide preclinical and early clinical materials to support phase 1 and phase 2 clinical trials, while the Flexible Biomanufacturing Facility, stand for FBF, is designed to provide not only clinical material from phase 1 to 3 clinical studies, as well as for product commercial launch. Both facilities have biosafety level 2 containment, with our new FBF facility has potential to go beyond biosafety 2 level containment. So among those 2 facilities, we currently have about 200,000 square foot GMP space, with 1,000 square foot space in NCTM building and another 1,000 square foot space in FBF buildings. Both facilities utilize the mobile clean room technologies. With first and second generation MCR currently installed in the NCTM building, while the third generation MCR stored into the FBF buildings. Mobile clean room itself is a fully segregated and self-contained unit. We can use VHP-based cleaning system in order to rapid changeover and return to neutral state after the completion of clients' programs. In addition, multiple MCRs can be connected to meet the program's need, such as upstream and recovery can be done in one MCR, with the downstream purification to be complete in another MCRs. Our Texas site has nearly exclusive use of single-use biotechnologies to support from cell banking to viral product GMP manufacturing. The site has a fully functional process development team, as well as analytical method development team with virology expertise in-house. We also have extensive quality system, which has released multiple lots of clinical viral and non-viral bulks. In addition, high-speed robotic viral product fill finish line is currently in place. Will be up running in will be up running in Q4 of 2018. Developing viral vaccine and gene therapy treatment sometimes can be challenging.